Welcome to this explainer. Today, we're diving into a fascinating microscopic assembly line. We're going to look at the exact, meticulous process that takes raw tissue straight from a biopsy and transforms it into a perfectly clear medical diagnosis. If you've ever wondered how pathologists can just look through a lens and confidently identify a disease, well, you are in the right place. So you might be wondering, how do we make invisible cells tell the story of a disease? It's a great question, because straight out of the body, raw tissue is incredibly fragile. It's practically invisible under a light microscope. You can't just slap a raw specimen on a piece of glass and expect to see cellular structures. Instead, we have to meticulously build a readable sample, step by step. We're removing water, adding wax, slicing it incredibly thin, and ultimately painting it with chemical dyes. Our agenda today covers 6 main steps. We'll start with conventional tissue processing, look at rapid microwave processing, the purpose of staining, direct versus indirect staining, progressive versus regressive staining, and finally wrap up with specialized staining. Okay, Section 1, conventional tissue processing. This brilliant circular workflow shows the exact path our specimen takes. Notice how it moves through graded alcohol, starting at 50% all the way up to 100%, just to completely dehydrate it. Then it moves into clearing solvents, like Xylene. Often vacuum pressure is applied right here to ensure the final component, which is molten paraffin wax, can fully infiltrate those delicate cellular structures. Now, there are 11 distinct technical steps in total here: fixation, decalcification, dehydration, clearing, impregnation, embedding, trimming, section cutting, staining, mounting, and finally labeling. It's a lot, right? But to make it easier, we can basically group this entire factory line into 5 major phases, from prep and fluid exchange all the way to waxing, slicing, and finishing. This overarching process is what transforms that raw biopsy into a preserved, readable slide. But listen, this entire process is absolutely fragile. In fact, ruined samples are almost always due to human error. If tissues are packed too tightly in their baskets, the fluids just can't exchange properly. The result? You get these fuzzy sections that are literally impossible to read. Or, if baskets aren't clean, wax accumulates and ruins the whole run. Technicians have to strictly maintain fluid levels to prevent sections from failing to stick to the glass later on, and they have to ensure tissue thickness is perfectly sized, ideally around 1.5 millimeters. Section 2, rapid microwave processing. So, what if a patient is literally on the operating table waiting for a diagnosis, and we need answers way faster than conventional batch processing allows? Well, by upgrading to microwave irradiation technology, we can cut tissue processing time by a massive 60 to 80%. It actually relies on the same heat diffusion principles discovered by Percy Spencer. You know, the guy who accidentally melted a peanut cluster bar with radar waves back in the 1940s. A lab can achieve an hourly throughput of up to 120 specimens this way, and microwave processing actually provides excellent morphology, sharper nuclear detail, and crisper overall cellular images. But to be clear, these are not your standard kitchen microwaves. These are highly controlled, sophisticated instruments. They're designed with thermocouple temperature probes to maintain heat within a very strict 1 degree Celsius range. They utilize molecular-friendly reagents, continuous workflow, and traditional vacuum infiltration to produce highly standardized, same-day diagnoses. And the best part? They often do this entirely without the use of harsh chemicals like formalin and Xylene. However, this incredible speed for sure comes with strict rules. The tissue absolutely cannot be thicker than 1.5 millimeters, or the reagents will penetrate unevenly. Also, paraffin has to be added in liquid form because microwave energy just won't melt wax pellets. And because it relies on flash evaporation, hitting very specific boiling points like 82 degrees Celsius for ethyl alcohol, continuous flow processing requires constant, hands-on attention from the operator to keep everything moving. Right. Section 3, the purpose of staining. Once our tissue is encased in wax and sliced incredibly thin by a microtome, it moves to the most visually striking phase. Like we touched on earlier, without stains, we are practically flying blind. After we remove the poorly permeable paraffin wax using solvents, we introduce dyes. These dyes are what finally bring the physical characteristics and structural relationships of the cells to life. They provide the contrast we absolutely need to definitively say whether a disease is present or not. Broadly speaking, staining is broken down into 3 major categories based on our target. We've got histological staining for basic tissue structures, histochemical staining to study various chemical constituents within the tissue, and immunohistochemical staining. That last one uses incredibly precise labeled antibodies, either fluorescent or enzyme labeled, to detect specific phenotypic markers.
Section 4, direct versus indirect staining. Let's look at how this builds into the actual mechanics of coloring the tissue. Direct staining is wonderfully simple. You apply a basic dye carrying a positive charge, and it's naturally attracted to the negatively charged cell walls and cytoplasm, sticking directly to the tissue. Indirect staining, on the other hand, well, it needs a little help. It uses a secondary component, either an accentuator to just speed up the reaction, or a mordant to physically link the dye to the tissue. Look at these puzzle pieces here. The tissue and the dye simply do not fit together on their own. But the mordant acts as a lock-in key middleman. A great example is potassium alum used with Ehrlich's hematoxylin. And fun fact, hematoxylin is actually a natural dye extracted straight from the logwood tree, whereas synthetic dyes rely on chemical structures called a chromophore and an auxochrome. Anyway, this mordant forms a colored lake, permanently bridging the gap and locking the tissue, mordant, and dye together into one cohesive complex. It's brilliant. Section 5, progressive versus regressive staining. Another huge factor here is how pathologists actually control the intensity of the color they're applying. With progressive staining, tissue elements are colored in a specific sequence for specific time periods. Once it hits that perfect intensity, it stops. No washing required. Regressive staining is the exact opposite. We intentionally over-dye everything so it's deeply, deeply colored. Then we perform a step called differentiation. Basically, we go backward by washing it with an acid solution to pull the excess color back out until the specific structures we want are perfectly isolated. Routine H&E stains commonly use this regressive method. Section 6, specialized staining. Finally, let's look at a few highly specialized techniques that medical technologists use to hunt down very specific cellular behaviors. First up is metachromatic staining. This is truly a bit of chemical magic. Imagine pouring a pure blue dye onto a tissue sample, but instead of turning blue, the target cellular substance chemically reacts and turns bright red or violet. Dyes like toluidine blue or crystal violet are famous for this, creating incredible contrast by shifting their own color based purely on what they touch. To make those details stand out even further, we use counterstaining. This is applying a completely different color to provide background contrast. For instance, using hematocylin to stain nuclei a deep purple and counterstaining the cytoplasm with eosin in shades of pink. And pathologists also use highly advanced specific stains to make specific architecture pop right off the glass. Things like Sudan IV or oil red O for lipids. For connective tissue, they might use Gamori silver, Van Giesens, Massons, or PTAH. For nervous tissue, Nissel or Luxol fast blue. And for bone marrow, they rely on Right Gimsa. But what if the cells are still alive? That is where vital staining comes in. Sometimes we need to see living processes like phagocytosis in real time. Intravital staining involves injecting a dye like carmine or India ink straight into a living body to color specific cell systems. Supravital staining is done on living cells immediately after they're removed from the body. We can actually use dyes like Janus green to watch living mitochondria at work. No way, right? It's amazing. It's amazing. It is absolutely incredible to consider the invisible, meticulous labor that goes into every single tissue slide. But before a finished slide even reaches a pathologist, it has to undergo a few final, crucial steps. We're talking slide ringing with sealants like DPX or Canada Balsam to prevent drying out and fungal growth. And of course, meticulous labeling following strict CAP and Joint Commission protocols, which requires 2 unique identifiers like the patient's name and their accession number. Because at the end of the day, this isn't just some complex chemical assembly line. It's a process designed to definitively protect a human life. Thanks for joining me on this explainer. And hey, the next time you hear a medical diagnosis, ask yourself, what microscopic journey made that answer possible?